Hello everyone. In this video you will learn about the second type of cell division, which is meiosis. Keep in mind that meiosis involves in the formation of haploid gametes from diploid gametes. You have to understand the definition of few terms to understand more about this topic, such as chromatin, thin, long, uncoiled structure of DNA, found throughout interphase. Chromosome, thick, shorter, and coiled structure of DNA, found throughout cell undergoes mitosis and meiosis. Sister chromatids. Sister chromatids is two copies of a duplicated chromosome joined by centromere, formed in S phase. Chromatid. A chromatid is one of two identical halves of a duplicated chromosome. Sister chromatids has two identical chromatids. Centromere. A region of chromosome that joins sister chromatids and has attachment site of spindle fibers. Homologous chromosomes. A pair of chromosomes of the same length, size, and carry same genes. One from mother and one from father. Synapses. The pairing of homologous chromosomes during prophase 1. Bivalent. A pair of homologous chromosomes lay side by side formed by synapsis during prophase 1. Tetrad. Homologous chromosomes consisting of 4 chromatids formed by synapsis during prophase 1. Chiasma. A point of non-sister chromatids are in contact during prophase 1. Crossing over. The exchange of genetic material between non-sister chromatids of homologous chromosomes during prophase 1. Crossing over may produce non-identical sister chromatids, which is, one of the chromatid is non-recombinant chromatid, without exchanged genetic material, and the other is recombinant chromatid, carrying exchanged genetic material. Well, now, let's continue our lesson about meiosis. Meiosis is a type of nuclear division which produced 4 daughter cells, each having half number of chromosomes as the parent cell. Meiosis occurs in gonad cells or reproductive cells to produce gametes in a process called gametogenesis. Meiosis can be divided into 2 main stages, which are meiosis 1, where the first nuclear division occurs, and meiosis 2, where the second nuclear division occurs. Meiosis 1 divides into 4 phases: prophase 1, metaphase 1, anaphase 1, and telophase 1. Meiosis 2 also divides into 4 phases: prophase 2, metaphase 2, anaphase 2, and telophase 2. In prophase 1, chromatin condense or thicken and shorten to form chromosome. Synapsis occurs, where the homologous chromosomes pair up to form bivalent or tetrad. Crossing over between non-sister chromatids occur at chiasmata. Nuclear envelope disintegrate. And centrosome containing centrioles move to opposite poles radiating spindle fibers. In metaphase 1, pair of homologous chromosomes or bivalents or tetrads arranged independently at the metaphase plate. Spindle fibers attach to centromere, specific at kinetochore, of each chromosome. In anaphase 1, homologous chromosomes separated and move to opposite poles. This is due to the spindle fibers shorten, and pull the centromeres to the opposite poles. Centromere moves first, pulling the homologous chromosomes apart. Each of the chromosomes still consist of sister chromatids connected at the centromere. In telophase 1, the chromosomes or sister chromatids arrived at opposite poles. The chromosomes uncoiled and lengthened to form chromatin, which becomes invisible again. The spindle fibers disappear. Nuclear envelope reform around each nucleus, which is now haploid or has half the number of chromosomes of the original parent cell. Cytokinesis also occur by cleavage furrow in animal cells or cell plate formation in plant cells, in the end of meiosis 1. This will produces two haploid daughter cells. Each chromosome still consists sister chromatids. The cells then enter meiosis II. In prophase II, behavior of chromosomes are: the chromatin shorten and thicken to form chromosome, and becomes visible. Each chromosome consists of sister chromatids, which is two chromatids joined at the centromere. The centromeres are attached to spindle fibers. At the same time, nuclear envelope and nucleolus disappear, and centrosomes and centrioles move to opposite poles. In metaphase II, the chromosomes or sister chromatids arranged themselves at the metaphase plate or equator of the cell. The sister chromatids of each chromosomes are genetically not identical, due to the crossing over in prophase I. In anaphase II, the breakdown of protein that holding the sister chromatids together caused the centromeres divide. The spindle fibers shorten, and pull the sister chromatids apart to opposite poles as two individual chromosomes. Centromeres moves first. In telophase II, the chromosomes uncoiled and lengthen forming chromatin, therefore becomes invisible again. The spindle fibers disappear. Nuclear envelope reforms around each nucleus, which is now haploid or has half the number of chromosomes of the original parent cell. Now, the chromosome is as individual chromosome. Cytokinesis then occur, by cleavage furrow in animal cells or cell plate formation in plant cell in the end of meiosis II. This will produces four haploid daughter cells that are genetically not identical from one another, and from the parent cell. The significance or importance of meiosis are: meiosis produces haploid gametes by having the chromosome number. Meiosis maintains the chromosomal number from one generation to the next generation. Because, when haploid gametes fuse during fertilization, the chromosomal number in the diploid zygote is always similar to the diploid parental cell. Meiosis can lead to variation. Because, first, when the haploid cells or gametes fuse at fertilization, there is recombination of parental genes. Second, during prophase I, crossing over occurs and produce recombinant chromosomes, where the sister chromatids is not identical. Third, during metaphase I, independent assortment occurs. The homologous chromosomes arranged independently at the metaphase plate, resulting in a random combination of chromosomes at the opposite poles when the homologous chromosomes separated in anaphase I. Here are details about independent assortment. Homologous chromosomes arranged independently at metaphase plate during metaphase I. Since they randomly or independently arranged, there are two possibilities arrangement. After meiosis I is done, different combination of chromosomes in each cells are formed. After meiosis II is done, genetically variation of gametes will be produced. So, there are possibilities combination of genes caused by independent assortment in metaphase I. This promotes variation of gametes. There are some similarities between mitosis and meiosis. The similarities are: both involved DNA replication before the process begins. Both type of cell division passes genetic information to the next generation. And both occurs only in eukaryotic cells. The differences are: mitosis occurs in somatic cells, meanwhile meiosis occur in germ cell of animal cells and in megasporocytes or microsporocytes of plant cell. In mitosis, synapsis of homologous chromosome does not occur during prophase, meanwhile in meiosis, synapsis of homologous chromosome occurs during prophase I. In mitosis, bivalent or tetrad does not form, meanwhile, in meiosis, bivalent or tetrad are formed during prophase 1. In mitosis, crossing over does not occur and chiasmata do not formed. Meanwhile, in meiosis, crossing over occurs between non-sister chromatids of homologous chromosomes at prophase 1, and chiasmata is formed. In mitosis, chromosomes, or sister chromatids align at metaphase plate during metaphase. Meanwhile, in meiosis, homologous chromosomes, or bivalent, or tetrad align at the metaphase plate of metaphase 1, and sister chromatids align at metaphase plate of metaphase 2. In mitosis, separation of sister chromatids occur in anaphase. Meanwhile, in meiosis, separation of homologous chromosome occurs at anaphase 1, and separation of sister chromatids occurs at anaphase 2. In mitosis, cytokinesis occurs once. Meanwhile, in meiosis, cytokinesis occurs twice. In mitosis, 2 diploid daughter cells are produced. Meanwhile, in meiosis, 4 haploid daughter cells are produced. Lastly, in mitosis, daughter cells are genetically identical to parent cell. Meanwhile, in meiosis, daughter cells are genetically not identical among the daughter cells, and to parental cell.